Have you ever wondered how brains evolved? Like, how did we get from animals such as sponges that don't have brains or any kind of nervous system, all the way up to animals like ourselves, dolphins or elephants with a massive cerebral cortex? I'm an assistant professor at Loyola University, New Orleans, and I've built and I teach a class called Brain Evolution as part of our neuroscience program. Now, as many of you know, I'm a scientist and I study autism from various evolutionary perspectives. In one of my earlier videos on the Cambrian explosion of autism genes that went surprisingly viral, thank you by the way, I talked about how a large number of autism susceptibility genes evolved in the earliest fish, which were the first animals to have true brains. Now, many of the people in the comments wanted to learn more about the major transitions in nervous system evolution. So that's what this video is for, and I hope you enjoy learning about this stuff as much as I do. First, in order to understand the pre-nervous systems of modern animals like sponges and a closely related group of animals called placozoans, both of which continue to retain some features that were likely present in their earliest ancestors, we first need to talk about our closest single-celled relatives known as the choanoflagellates. Choanoflagellates are a type of single-celled eukaryote. Eukaryotic meaning that they have a nucleus, unlike other organisms like bacteria or archaea. Now, these little eukaryotes look sort of like a sperm, but with a collar underneath its cell body. Its tail is known as a flagellum, which helps it swim around. It can be either single-celled, or sometimes they live in colonies together. And they trap food in these collars and then consume the food via a process called endocytosis, which is where a portion of the cell membrane starts to invaginate and envelop the food, bringing it into the cell. Now, if we look at this diagram of a modern placozoan, we can see the cells lining its underside still look very much like the single-celled choanoflagellates. You can also see that the cells lining the inside of the sponge, which is basically its primitive gastrointestinal tract, also look suspiciously like choanoflagellates. So the basic idea is that some choanoflagellates that formed multicellular colonies eventually evolved into these earliest animals, which today are represented by sponges and placozoans. It may be a little harder to picture how a group of single cells evolved into something like a modern sponge because there were undoubtedly many more evolutionary steps in between, but it's a little easier to imagine how choanoflagellates evolved into a placozoan, which honestly I think kind of look more like a blob. All that being said, hopefully you can start to see that the choanoflagellate cell type is the basic building block of these primitive animals. In particular, those tail-like structures help these modern animals with movement, sensation, and digestion, which are still basic themes present in our own bodies today. And in fact, many of the sensory-related hair cells in our bodies, such as the ones involved in hearing and your vestibular sense inside your inner ear, as well as the hair cells that have been morphed into photoreceptors in your eyes, all of these get their basic cellular body plan from our little choanoflagellate ancestors. Okay, so even though sponges and placozoans don't have anything we would call a nervous system, they are nevertheless capable of rudimentary forms of movement, sensation, and digestion. What's more, many of these choanoflagellate-like cells in these animals do express nervous system-related genes, such as various synapse genes. That means that important groundwork was being laid that later animals developing actual nervous systems could take advantage of. Now let's move on to the next two groups of modern animals that represent the next stage of nervous system evolution, and that's the ctenophores and cnidarians. The ctenophores include animals like comb jellies, while the cnidarians include animals such as jellyfish, sea anemones, and corals. Now we're grouping these two clades of animals together, even though they're only distantly related to each other, because they both have the most primitive style of nervous system in modern animals, and that's something called a nerve net. Like the name implies, these animals have a net-like structure of nerve tissue running through their bodies. They also have some clusters of neurons in certain regions associated with simplistic sense organs. But for the most part, most of the neurons are diffuse in these animals and form net-like structures. In addition to these nerve nets, ctenophores and cnidarians also have primitive muscle cells known as myoepithelium, which is a muscle-like tissue that's less similar to your skeletal muscle and more like the muscle that lines your blood vessels and intestines. Ctenophore and cnidarian ancestors were also the first animals to have true sense organs. For instance, the ctenophores have what's known as an apical sense organ that senses gravity, helping them to tell up from down and know where they are in that water column. Meanwhile, the cnidarians have a structure called the rhopalium, which also has cells that sense gravity, as well as other cells that sense light and chemicals, providing rudimentary senses of sight and smell. The next group of animals we're going to talk about are the bilateral animals. Now, if we look at sponges and placozoans, their bodies don't have any symmetry. Meanwhile, ctenophores and cnidarians are radially symmetric, meaning that they have multiple lines of symmetry, all of which intersect at a central point within the animal. But bilateral animals only have a single point of symmetry, right down their midline, creating mirror halves and allowing you, metaphorically, to fold them in half onto themselves. Every animal, from a fruit fly to human, is bilaterally symmetric. Now, why do we care about symmetry? For one, that's how we define bilateral animals, so it's important we know which animals we're actually talking about. The other thing, which is more important for the purpose of this video, is that the nervous system becomes centralized in bilateral animals, running right down the center of the body from head to tail. We also start to see larger groupings of neurons called ganglia, especially within the head region, thanks to some of the sensory organs that are evolving there, like sight, the vestibular system that processes balance, and smell. A centralized nervous system, or CNS, is really important in bilateral animals because it's a major center for integrating incoming sensory information and then coordinating body-wide responses like movement. By contrast, the nerve nets of ctenophores and cnidarians only receive and process sensory input and determine motor output locally, so sensory and motor processing are much less coordinated than we see in the bilateral animals. Now, why do nervous systems become centralized in bilateral animals in the first place? Well, one of the likely reasons is the mode of movement that bilateral animals use compared to non-bilateral ones. The nerve cord closely appends the region where the muscles of the back and trunk, including the legs or fins, meet along the midline, suggesting that a major reason for the change in nervous system structure was a change in the way these animals moved, notably moving forward with the head leading and often utilizing limbs or trunk, in the case of our ancestors, in an alternating right-left coordinated motion. So it probably has a lot to do with the way we sensed and moved through the ocean more than 600 million years ago. Now, the next major leap forward in our lineage occurred around the time of the earliest fish, aka vertebrates, which is anything that has a vertebral column made of bone or cartilage. The oldest primitive fish fossils that we found date to around 530 million years ago during the early part of the Cambrian period. These are animals like Hycuichthys and Millicommingia. As I discussed in my 3-part series on the Cambrian explosion of autism genes, the early vertebrates experienced 2 whole genome duplications, which led to a massive expansion in their genetic material. This sudden increase in genetic material is linked with an expansion in the size and complexity of the nervous system, and in particular is associated with the evolution of a true tripartite or 3-part brain. This is the brain archetype or blueprint that all of us vertebrates have, and it's something we share with our earliest fishy vertebrate ancestors. All other aspects of brain evolution in vertebrates are a variation on this deep and well-entrenched theme. Now, if we fast forward, we can see that all the basic brain building blocks are there, with the exception of the cerebellum, which is that little a mini brain kind of towards the back of your head. That evolves later in jawed fish. But even early fish like Hycuichthys and Millicommingia are thought to have likely had a rudimentary telencephalon, which is the region of our brains that the massive human cerebral cortex develops from. So the fundamental pieces were all there, and they've been slowly tweaked to form the zebrafish brain, the lizard brain, the bird brain, the mouse brain, the dolphin brain, and the human brain. Now, in my class, I explore more about some of the evolutionary changes in the brain across terrestrial or land-dwelling vertebrates like the amphibians, mammals, primates, and the human lineage. So if any of those topics interest you, just let me know in the comments and I'll add them to my video to-do list. Well, thanks again for joining me down probably one of my favorite rabbit holes, deep brain evolution. Do please be sure to like, subscribe, share, and let me know what other questions you may have about the brain, evolution, and autism. As you know, I always try to respond to each of you. So until next time, take care and ciao.